Welcome to this video on cellular biochemistry and the evolutionary history of life. We will explore energy-producing enzymes, the complex machinery of polymerases, the three domains of life, and the fascinating origins of endosymbiotic bacteria. In this first section, we dive into energy-producing enzymes. These are the vital catalytic molecular engines that drive metabolic pathways and are responsible for the generation of ATP in cells. Cells primarily generate energy through two methods: substrate-level phosphorylation, which occurs in the cytosol and is independent of membranes, and oxidative phosphorylation, a high-yield process driven by proton gradients across membranes. ATP synthase acts as a remarkable molecular rotary motor. Driven by proton flux, it rotates to undergo mechanical conformational shifts that synthesize ATP from ADP, an inorganic phosphate with near-perfect efficiency. Three core complexes power this system. Glycolytic kinases break down glucose, Krebs dehydrogenases harvest high-energy electrons, and cytochrome oxidases consume oxygen to maintain the necessary proton gradients. The efficiency of these processes varies significantly. Oxidative phosphorylation is vastly more productive than fermentation, yielding up to 15 times more ATP per glucose molecule compared to substrate-level processes. In our next section, we turn our attention to polymerases and macromolecular synthesis. These specialized enzymes are essential for constructing the genetic code of life. Polymerases are complex proteins that act as nucleotidal transferases. They facilitate the nucleophilic attack required to form phosphodiester bonds, using the energy released from pyrophosphate cleavage to drive synthesis forward. Polymerases feature a distinct right-hand domain structure that allows them to grip DNA with precision. They ensure accuracy through template-directed matching and proofreading, while RNA polymerases natively unwind DNA to synthesize transcripts. The central dogma relies on three primary polymerases. DNA polymerase replicates genomes, RNA polymerase transcribes genetic sequences, and reverse transcriptase synthesizes DNA from RNA templates, a common feature in viruses. Polymerases operate at incredible speeds and fidelity. DNA polymerase III can incorporate 1,000 nucleotides per second, supported by sliding clamps that ensure processivity and high fidelity through rigorous proofreading. Our final section examines biological lineages. Understanding these fundamental divisions of life requires looking at distinct biochemical markers. Carl Woese's research revolutionized our understanding of life by identifying three distinct domains: bacteria, archaea, and eukarya. Eukarya are characterized by their complex, nucleated cells containing organelles derived from ancient endosymbiotic events. This matrix compares the three domains across several features, including membrane lipid chemistry, cell wall composition, RNA polymerase structure, and the presence of histones, which clearly delineate these unique lineages. Finally, archaeal membranes are uniquely stable due to their ether-linked lipids. Furthermore, genomic studies reveal a deep homology between archaeal and eukaryotic transcription machinery, placing the origins of eukarya within the Asgard archaea clade. Moving to section 4, we examine the ancient parasitic endosymbiont, exploring how a bacterial infection transformed into the powerful eukaryotic engine we know today. Around 1.8 to 2 billion years ago, an anaerobic archaeal host internalized a free-living, aerobic alpha proteobacterium. This relationship, possibly starting as an invasion, evolved into an obligatory metabolic mutualism known as endosymbiosis. The process unfolded over millions of years. From the initial engulfment during the Great Oxidation Event to metabolic syntrophy, endosymbiotic gene transfer, and finally, the complete organelle lock-in, this timeline shows the integration of the mitochondrion into eukaryotic life. This integration brought massive changes. Most bacterial genes migrated to the host nucleus, leaving only 37 essential genes in the mitochondria. This yielded a huge bioenergetic surplus, fueling genomic expansion and complex multicellular life. In summary, life's complexity relies on three pillars: energy-producing enzymes like ATP synthase, accurate polymerases for genetic transcription, and the ancient endosymbiotic mergers that provided the energy for life to flourish. This concludes our video on cellular biochemistry and evolutionary lineages. Thank you for learning with us about the fundamental mechanisms that drive life on Earth.